So with the N9831 clinical trial, it is a pivotal trial that used Trastuzumab in the adjuvant setting. This particular trial had three arms. So the first arm was the chemotherapy alone arm. Arm B was the arm that treated sequentially between chemotherapy followed by Trastuzumab, and the last arm was the concurrent chemotherapy arm, concurrently between chemotherapy with Paclitaxel and Trastuzumab after anthracycline, like AC chemotherapy. So we did the gene expression analysis in this particular study using Oncotype Dx assay and also PAM50 assay. So what we found was that in the patients with HER2 positive breast cancer that treated with adjuvant Trastuzumab, when you look at the risk of early recurrence within the first 5 years, gene that associated with the ER signaling and ER axis were associated with improved outcomes. So patients with ER positive disease tend to have less recurrence within the first 5 years, which we actually see in the clinical practice. Genes that related to the invasion axis, like in the Oncotype Dx and also Oncotype risk group itself, if you divided the Oncotype into three risk categories according to low, intermediate, and high, patients with higher risk group seem to have worse outcome with the early recurrence, and also patients that were classified as basal, like subtype based on the PAM50 gene, those patients also had early risk of recurrence in the first 5 years. On the other hand, if you look at the risk of late recurrence, unfortunately none of the genes seem to be associated with the risk of late recurrence. So that is something that's still unmet need, and that's something that we need to try to find out which are the predictor of these patients with HER2 positive breast cancer that are still at risk of late recurrence after 5 years of therapy.